Okay. Hello, everyone. My name is Mauricio Monroy. I'm the actual president at Quanta, the student group that is dedicated to science topics and to make science fun. Today, I'll leave you in the capable hands of Meje Cauja, who will have the power of the forum. Good luck. Have fun.
Hello, everyone. I would like to present our presenter today, which is a chemistry teacher, our chemistry teacher from Campus Esmeralda, Olatunde Oparemi, and he will be presenting plant tissue culture today for us. So I will leave the talk to him.
Thank you so much, Mauricio and Mehek. I'm very honored to be with you today. I've known most of the faces here: Julia Estudillo, Daniel, Diego, Ivan, Bobby, Cano, Shirel. Yes, who else? Lore, Vale, Henry, and of course the rest. I'm very glad to be here today. So I'm going to be talking about plant tissue culture today. I'm going to be sharing my experience. I had the opportunity of participating in plant tissue culture programs, and I'm going to be talking about the benefits, the processes, and the future possibilities that this technology has, especially that we live in a polarized world. So, and there are a lot of things that that may not directly affect us, but that we may be taking important steps in making this world a better place for all of us. So most especially as science students. So we should be thinking of how to be global citizens and how to create an enabling world, a much more sustainable world for everyone. So I'm going to, I want to be sure if everyone can see my presentation. I'm sharing my screen. Can everybody see my presentation? Yes?
Yes, sir.
Great. Thank you so much. So thank you. So I'm going to, just like I said, I'm going to talk about my experience on plant tissue culture, and I'm going to try to explain the basic knowledge and the basic process so that we have a general idea of what this is all about. And then as students in high school, when we are making decisions to see what we can study in the future, where some of us may be interested in, choosing a career that is, that is similar to these in the future. So I'll be sharing my knowledge. I'm not very well prepared today. I would have loved a situation where I'm much more prepared, but I'm going to try my best to explain as as clear as possible. So I want us to please take a look at these three plants, and I want any of you to tell me if you know any of them, and perhaps you know any plant that is similar to all of these three. Yes, anyone? Does anyone know any of these three plants? Anybody? Yes? Is anyone talking there? Babi Cano? Karen Hernandez? Yes? Anybody? Has anyone seen any of these plants before? Okay. So the first one is called Calamites. I don't know if you know anything about this plant. It's one of the ancient plants. Why this one in the middle is the Saint Helena olive plant. So this is also used to be very important plant, and then the last one is one of the primitive plants. It's the last. In fact, right now, it's even difficult to find the fossils of these plants, of these Cooksonia, because all of these plants that I'm talking about that we have on our screen right now, we can't find them anywhere else, anywhere on Earth today. We can't find any of them. Something must have happened to them. Perhaps what happened to the dinosaurs. Okay, so let me give you a practical example here. In Nigeria, this is called moi moi. In Mexico, we have something similar that we call tamales. But the basic difference is that this moi moi is made with beans. Tamales is made with with maize. So, but this is made with beans. And there's a special plant, the plant leaf that they use to wrap this in the process of cooking it. As you can see here, they are using a special leaf to wrap it in the process of cooking it. That leaf gives a special flavor to the moi moi to make it much more delicious, you know. However, something is happening to this plant. And I was able to participate in the process of regeneration and, you know, conservation of this plant. Something is happening to it. What happens to the 3 previous plants that I just showed you is actually happening to this plant. So the leaves are no more available to make moi moi. In fact, they are using something like this instead of the leaves. So the culture of eating the moi moi with the plant flavor from the leaf that they use is disappearing as well. So why am I saying all this? I'm giving this introduction in order for us to know the importance of plant tissue culture. All of these plants that we've talked about, including these leaves, the plant is called no, I've forgotten the name. Thamomyces daniela or something. I've forgotten the scientific name, but the specific name is Danieli. Why the generic name is Thamomyces or something, I've forgotten it. But actually, this plant and the previous one that I've talked about, they have something in common. The previous one I talked about, they've gone into extinction. The one that you can see on your screen is on the verge of extinction. So where does tissue culture comes in? So let's quickly talk about what tissue culture is all about. Then we can link everything together and see how tissue culture might be the solution to some of the problems we have in the world. Because most of the time, people focus on extinctions of animals, forgetting the fact that everything depends on the plant. They are the primary producer in every ecosystem. So, which means we should also not only focus on the extinctions of the animals, but also the extinctions of the plants as well. And also there are more possibilities that tissue culture can give us in order to make this world a much more sustainable world. So let's talk about tissue culture itself. I don't know if any of you have seen people growing plants in test tube before. So this is a test tube. You can see plants growing in the test tubes. So which means plants are being grown in vitro in the laboratory. So the process of tissue culture actually refers to aseptic growth of plants or plant parts in the laboratory using appropriate culture media. So, and something that also goes with the definition of tissue culture is that you are producing clones. Imagine I can clone mehek. I can make 1,000 of mehek. The same mehek, imagine that. That would be amazing. So the same height, they say everything. They speak the same, you know, that would be cool. So the process of plant tissue culture refers to the process of cloning plants, producing plants which have the same genotype, the same genetic constitution under aseptic conditions. So when we take a look at this, the beauty of this is that we are producing the plants under a condition, aseptic condition. So for those of you who are studying biology, IB biology, or who are exposed to people who are in the microbial field, or the microbiology field, or biotechnology field, you understand the concept of aseptic. When they culture something or when they grow something under aseptic condition, it means it's disease-free, no infection at all. So the process of tissue culture, this is exactly what we do there. So I have the opportunity to work in where we call National Center for Genetic Resources and Biotechnology. And I have the opportunity to work with them in the 2 basic programs that I'm going to be sharing the experience with you in a moment, so that we can have the basic understanding of plant tissue culture. And when some of you want to make the decision of what to study in the future, you may be considering these as an alternative for you, as the best alternative for you, because there are a lot of possibilities out there that have not been exploited at all. People are not even talking about this. And there are a lot of possibilities. So let's talk about the principle of plant tissue culture. Plant tissue culture is based on the principle. It was actually it's not new. You know, people have done this before. Yes. The basic principle is based on the fact that plant tissue have this property that we call totipotency, you know, which implies that a single living cell can actually develop or regenerate into a complete whole organism with the same phenotype. So when enabling environment is provided, in fact, we've seen, for those of you who have done agriculture, maybe in the secondary school, in your junior high school, you've seen what they call method of propagation, plant propagation. Some plants, they can cut the stem, they put it in the soil and it grows to a completely new plant. There is one plant, I have forgotten the name, brio something. That plant, just cut the leaf. The leaf alone, put it on the soil. The leaf will form adventitious roots and it will grow to a complete plant. But that is different. Those occur in vivo. But we are talking about in vitro manipulation or growth of plant parts. So, but the basic principle is based on the fact that there are some parts of a plant that can grow into a complete plant part and can, you know, that we have the same genetic constitution as the plants. So, which is actually basically cloning. So it's like cloning plants. So plant tissue culture is basically cloning plants. So let's talk about the materials and equipment that we use in this process. The first thing we talk about the explants. We have the glassware. We have the growth medium, which is the most sophisticated part of this, which I'm going to be talking about each of these. The aseptic condition, yes. If the aseptic condition is not provided, everything can be infected. So aseptic condition is highly needed. A culture room is needed. An incubation room is also needed. There's a difference between the two. In most cases, they use the culture room as the incubation room as well, but the two are two different things. So I'm going to be talking about the first one, which is the explants. What are explants? What exactly are explants? In order to carry out a successful tissue culture or plant tissue culture, the first thing you need to think about is, where should I get my plant material? First thing, they must analyze the plant that they want to culture. Let's say Mauricio wants to culture a very attractive, colorful, a plant that has beautiful flowers, and he thinks that this plant is not available in Mexico. Maybe he saw the plant in India. He said, "Oh, he wants to bring the plant to Mexico." He say, "Okay, but the conditions, the weather conditions will not let the plant grow. Can I grow it in vitro?" Oh, yes. Then Mauricio will say, "Okay, what can I do? What part of this plant can I use?" So the choice of explants is actually based on years of experience, years of working with them. You must have tried the different part of the plant to know which one works best. But every part of the plant is vital. It can be the leaves, the auxiliary buds, the internode, even the stem, root in some situations. So that part of the plant, the tissue, the plant tissue, the small piece that you want to cut away from the mother plant and put in the test tube and make it grow under sterile or aseptic condition in the laboratory is your explant. So the first thing we do is exploration. When we say we want to do exploration, we go into the wild, we go into other research institutes to look for the explant of the plant material that we want to culture. So that is the first thing. It can be very tedious. I remember when we wanted to culture a plant known as Artemisia. Artemisia is a plant that some people claimed cured HIV/AIDS. And also, some people claim that it actually works for malaria, as we know that malaria is a very serious problem in African countries. So it kills people, millions of people every year. So we actually wanted to get that plant. We couldn't find it anywhere. So some people were asked to travel to Ghana, and some were asked to travel to England to get the explant for the plant in order to propagate it, culture it, and transfer it to the field so that we can have a lot of pieces of the same plant so that we can use it for the experiment that we wanted. So explant is the first thing. Getting your explant is the first thing. Is a vital process as well. Then there are some basic equipment that are very important. You cannot just do tissue culture. In fact, these photos are the original photo I took then. I asked my sister to send some of those photos from my computer back in Nigeria, but she didn't send them to me on time. So for example, this one, I remember this one is actually the autoclave, the autoclaving machine that we used. It's a very old one, very old and very big, but it worked really well. This is the autoclave for sterilization of equipment that we use at a very high temperature. This is another one, very small one. This is like pressure pots that people use for cooking frijoles, beans. So it's like a pressure pot. But this is the old autoclaving machine that we use. There are much more beautiful autoclave smaller versions of this right now. There are a lot of those ones right now. This is the pH, the digital pH machine. This is the weighing balance. This is the oven. A lot of test tubes are needed. You are going to use a lot of distilled water. So you are going to need a distiller as well. These are the basic equipment that are needed for successful tissue culture. In fact, most of you in your respective schools, I mean the professional school, the university in Campus Ciudad de Mexico, the university in Santa Fe and in SEM, I think they have a lot of equipment, state-of-the-art equipment that you can actually, ask for permission to carry out some of these processes in their labs. They have a lot of beautiful equipment there that you can take advantage of. So you just need to write a letter. If you want, you can. If you want me to help you, perhaps you want me to actually be part of it, you can let me know. So these are the basic glasswares and equipment that we use in tissue culture laboratory. So let's talk about the most difficult part. The key to success or to a successful tissue culture depends on the media. So I'm talking about the media. You are looking, where's CNL? Where is Azteca Una? So the media I'm talking about here is like, I don't know if you can see this. This is mole de olla, I took this mole de olla to school, but I didn't have time to eat it. So I brought it back home. So what I'm saying is that the media is like the food. Just like this mole de olla is to Olatunde, the media is to the plants. It's like the food that the plants eat. For those that culture microorganisms as well, the media is necessary. A specific medium is necessary. So the culture media, the preparation of the suitable culture media is the key to a successful tissue culture. So no, the medium of on which we culture the plants are not the same, even though they have some basic components. Every media must have inorganic salts, the macro and the macronutrients. We you know, the macronutrients. We talk about the NPK, NPK, the nitrogen, the potassium, the phosphorus, calcium, and what have you, and the micronutrients, iron and what have you. All these must be present. These are basic for plants, effective and healthy plants growth. So the carbon source, remember, I don't know if you have done Krebs cycle, if you have done these, what is it called? Photosynthetic pathways. I'm not even talking about Krebs cycle. I'm talking about what do we call the carbon cycle and Calvin cycle? Yes, the Calvin cycle, or all these, you know, the carbon source is essential. Plants use the carbon source as energy source. So in this case, glucose is essential in all the media. Then we have vitamins, also some growth hormones like auxin, cytokinin, and what have you are added to stimulate roots development, shoots development, and what have you. So the gelling agents, and of course, there are other natural complexes that must be present. So the key to any successful plant tissue culture depends on the media. So in fact, I was lucky at the time I was in National Center for Genetic Resources and Biotechnology. There was a woman who was a specialist in media preparation. So I was able to work with her to see how they prepare media. So right now, if you want to make your own tissue culture, you don't need to prepare the media. You just buy it from the market and try which media works best. It's just like doing the scientific experiment. You can set it up. You can buy this media, these and that. You can have 3 different media, put the same type of tissue culture on them, and see which one makes your tissue grows best. But actually, the development of the media is a much tedious process because you need to take into consideration a lot of the physiology of the plant. You know, you need to know, is it a plant that requires a lot of fertilizer? Is it a plant that requires a lot of nutrients? A lot of, you know, some plants are low light requiring plants. Some plants are low carbon requiring plants. Plants are very different. So some culture media are very specific to the plants that you want to culture, that you want to grow. But some are generally used for many plants and they work for many plants. So the media selection is very, very important, and preparation. When preparing your media, it must be 100% aseptic. If not, bacteria can grow on it and fungi can grow on it, and that will infect your culture. So these are the basic one. We have the hemos medium, B5 medium. We have the whites medium, developed by different scientists, and the basic differences in them are some of the percentage of some components that they have. But the basic component, just like I've said, the carbon source, the macro and the micronutrients, the vitamins, and then as well as the gelling agent. So all these are very vital when talking about the media. So we've talked about, the first thing we talked about, can anybody remind me? The first thing we talked about? Anybody? A volunteer there? Yes, Julia Estudio? Yes? Yes? Julia?
Explant.
The explants, as Ivan Aritpe, thank you so much. The explants. The explants are very important, you know. You need to explore. You need to go on exploration to get the plant parts that you want to culture, yes? Then you need to have the appropriate equipment. You need the culture room, the inoculation room, the glasswares, the autoclaving machine, autoclave for sterilization of the equipment and what have you. You need a distiller for distilled water. If not, you need to get a large quantity of distilled water. to be used in the process. And of course, media, the cultural media is another one. Thank you so much. Thank you. Then the inoculation and culture room. Yes. So this is where we inoculate. In fact, I got this photo, this one, the photo on my right, this one. This is not from me, but this one, the one on my right, I got it in one of my drive because we were making fun of this girl at that time. Her name is, we call her Babi. Anu. So she was culturing and she was shaking. That was her first time inoculating, you know. That was her first time inoculating. And then, so we took a photo. She was so serious. She was never serious. She was a very fun, loving person and always playing. A very happy person. But in this situation, she was very serious and very, but she was shaking. So we took a picture and we were making fun of her. But eventually she did a lot of work and she was very successful in it. So the inoculation room, this is where we do the major work. Everything must be aseptic. People normally don't wear gloves here because before you go in, you sterilize your hand. You sterilize your hand. You surface sterilize the place. Everything is, one thing that is important that she's not doing is that she was not wearing mask. Is very essential because microorganisms are ubiquitous. You know, by the time you take a deep breath, you are infecting the culture media. So you know what, you can see all these people here, they are wearing nose mask, but they are not using glove either because they must have sterilized their hand. In fact, in a good inoculation and culture room, when you arrive, the first thing is to wear the lab coats. You sterilize your hand, you remove your shoes or whatever you have. There's a special kind of shoes or is a wood, like a cloth, but for the feet. You wear it on your feet, then you sterilize your hands again. You wear the overall or the lab coat, and then you surface sterilize the place. You wear your nose mask. So the process is very, very tedious to make sure that your tissue culture is successful. So then the inoculation room is essential as well. It can be done in the wood. I think we have one in Prepatec Esmeralda, Ms. Marta. She's in charge there. She's always helping us to make sure that we take maximum advantage of the lab and also provide us with the basic equipment that we need there. So we have one in Prepatec Esmeralda as well, where you can have the hood, you can, and then the beauty of this is that you don't open everything in order to minimize the amount of hair or the exposure of your culture and the explants to hair. So you want to minimize that so that you want to make sure that the condition is as aseptic as possible. Then we also have the culture room. This is another one. The culture room here, as you can see, this is where we keep the plants after they have been inoculated. After the explants have been sterilized, you need to sterilize. I'm going to talk about this process. I'm just talking about the equipment now. So this is the culture room. This is where we put the plants. You allow them to, like 16 hours of light to simulate photosynthesis. So, the natural light outside. So you need to supply them with light. So, and then you need to control the humidity and what have you. So the culture room must be aseptic, highly aseptic, no contamination, clean, and beautiful. So yes, let's see. Let's talk about the process, the stages in plant tissue culture. So we've talked about the materials that is needed. We talked about the process of tissue culture itself, which is literally cloning plants. Yes. So the first is when we have all the materials ready. So the first thing we do, we prepare our nutrient media. You can buy it in form of powder. So you prepare it. You mix with it adequate number and amount of distilled water. You weigh the correct amount of each of the components to be added together. Then, you need to prepare the media. That is the first thing. Then selection of suitable explants. And after the explant is been selected, because I talked about exploration and selection of explant before, then we need to talk about the sterilization. You need to surface sterilize the explant. We normally soak the explant, the plant material, in bleach to kill all the bacteria or microorganisms that may be present, because you don't want to infect your culture. And also, something important is that the duration of the time varies. If you are taking the plant from the wild, it is recommended that you soak it up to 20 or 30 minutes. But if you are taking it from a very beautiful garden, or in the case of a plant taken from a glass house, a greenhouse, sorry, or a screen house. I'm going to talk about the difference between the greenhouse and the screen house in a moment. So, they are less contaminated, so it may take less than that, but we usually soak them in sodium hypochlorite for 20 minutes. And then once our explant is surface sterilized, we inoculate the media. We have the culture media, which we usually put in test tubes. Then we are going to cut the pieces of the plants that we need, maybe the node, the piece of the leaves, or any plant culture, suitable ones, based on the experience that we need. Then we are going to aseptically transfer it into the culture media, just like growing bacteria. Boom. Yeah, aseptically transfer it into the test tube. Then you are going to incubate. You are going to incubate it in the culture room. You are going to put them in the culture room at a temperature of at room temperature, relative humidity of between 50 and 70%. Then 16 hours of light and 8 hours of light and darkness. So in the culture room. So it's a very illuminated place, and everything must be under aseptic condition. So within few days, you will see, within 5 days maximum, the little material, the little plant material that you put in the test tube will start to grow. Depending on the kind of plant, some even grow within 24 hours, will start to grow. Take the nutrient from the culture media or the nutrient media and grows, beautifully grows. And once they grow in test tube, let's say I have 10 test tubes, there's something we call regeneration. We can actually use the one that grows under aseptic condition, no disease, nothing. We can now cut them into pieces. So we cut them into pieces, we re-inoculate more. Let's say I have 10 test tubes and I'm cutting, after they've been incubated, they grow. Now we can cut each of them into three pieces and introduce them into different media again. So which means I'm having more. It's a very tedious work, because sometimes we were given the mandate to produce 1,000 plantlets of banana when I was working in this 1 million, not 1,000, 1 million. 1 million plantlet of banana when I was working in National Center for Genetic Resources and Biotechnology. I went there for an industrial attachment. I went there as a student, so I worked there for a while. But it was a very beautiful experience for me. And then when they have all grown in the lab, the next thing we do is the process of acclimatization. I know that most of you have traveled abroad. You've been to Russia, Australia, or Canada. Canada is very close. You know how cold it can be in the winter. You know, it takes time for you to get accustomed to it. The same with these plants. When we bring them from the laboratory, you cannot just take them directly to the field. They die. They won't be able to withstand the heat. They won't be able to withstand the sunlight. The pressure in the natural world. So we need to acclimatize them. So what we normally do is to put them in nylons like this. We can put many together, and we are going to spray water in the nylon, tie it, and put them under a tree so that they can be exposed on a shaded trees. They can be exposed to light little by little. They can get acclimatized. And also, we are not going to change the media for soil directly. We normally use a special preparation. Coconut, we know the cocoa. They will say this que saben cocoa, the coconut. You know that, you need to peel off the I don't remember, the shaft and what have you, in order to get your cocoa, the coconut. So all those, we use this shaft and this material to prepare, because it's high in fibers and nutrients, and very soft as well. So we use it in the preparation of the soil, or in the preparation of the materials on which we transfer our plants that we got from the laboratory too. So this is very, very important. So the next thing, also the process of acclimatization can also be done in the screen house, in the screen houses. Or once they are acclimatized, we do not take them to the field directly. We take them to the screen houses. Greenhouse is these agricultural houses with glass on top. You must have seen them with glass on top. But due to the effect it has on increase in global temperature, average global temperature, they call it greenhouse gases, because when you are in the greenhouse, it's hotter than when you are outside of the greenhouse. So, this is a greenhouse gas because it traps the solar radiation, comes with a very high wavelength, passes through the glass, enters the greenhouse. Some of these radiation are absorbed by the plants. Some of them are absorbed by the concrete, and some, by the time they pass through two media, for those of you that are doing physics, there'll be changing wavelengths. The wavelength will be decreased. So the wavelength of the radiation will be decreased. And when they are reflected back, the wavelength, the wavelength will be long. The energy of the wavelength will be decreased, but the wavelength will be longer. So the wavelength will be longer, the radiation will be longer. We have longer wavelengths, which have weaker energy. They will not have enough energy to pass through the glass to escape into upper atmosphere. So more heat will be trapped in the glass house, so which makes it warmer. So because of that, people are no more using glass house. They use screen house with net around it. When you travel going on the way to Puebla, you see a lot of glass house and screen houses. So we keep them in the screen house. But the beauty of keeping these plants in the greenhouse is that you can see plant of the same size, the same number of leaves, green, everything, beautiful, no diseases. And you can have hundreds of thousands of plants in the small space. You can imagine that. So you save a lot of space. So that is the beauty of this process. And also you can see this is actually this is actually an image of the plants that I worked with, and these as well on the field. So you can see here, we have lot of plants of this with the same features in the greenhouse and in the screenhouse, and we transferred them to the field. We took them to the field. So once we have seen that they've acclimatized, they can now live under the natural condition, then we take them to the field to plant them for growth, and and then so that they can continue to grow under natural conditions. So you can see here, from here, we are going to remove the planting bags and transfer them into the field. So this is the basic of the plant tissue culture. This is the basic. This is the basic of the plant tissue culture. So my questions to all of you is, why is plant tissue culture important? Because that is the idea. Remember at the beginning, I was showing you guys some images of some extincted plants and the plant that is on the verge of extinction. So those are some of the examples of where we might be thinking to apply the knowledge of plant tissue culture. So I want you to please tell me, why is plant tissue culture important? Anybody? Volunteers? Yes? Go ahead. Yes, Mauricio, please go ahead.
Just to conserve species and to help prevent extinction.
Oh, that's a very beautiful one. Thank you so much. That's a wonderful one from Mauricio. Thank you. Thank you, Super Mau. That was a very good one. So it may be the alternative that we need in order to conserve species and to prevent extinction, as excellently said by Mauricio. So, which means we may be thinking of applying the knowledge of plant tissue culture and look for plants that are on the verge of extinction and try to culture them. You know, many factors are contributing to extinctions of plants and animals. The drastic change in weather, pollution. Some plants are only found in specific places because of the nutrient and the type of topography and, pH and what have you of the environment. So which means if we can take these plants, culture them, propagate them, multiply them in millions, which means we are going to save those plants, and we can actually think of, acclimatizing them to different conditions and save them from extinction. This can also be applied to the desert. Yes, someone to save time and resources as well. Oh, Henry, that's a beautiful one from Henry. To save time and resources, that's a very beautiful one, because we waste a lot of time and resources in planting, even the land. A lot of land is, in fact, look at the city here. Look at Mexico City, where I am. You can hardly find, space to plant. So which means we can maybe find a room in my house and put hundreds of plants in test tubes, in vitro, under aseptic conditions. Yes. So does anyone want to add more to what Mauricio and Henry said? Anyone else? Yes? A volunteer? Anyone else? Yes? No one else, okay? So apart from the fact that tissue culture might be an option for saving a plant species, it also helps to eliminate diseases from plants, because we do it under aseptic condition. It's also, just like Henry said, is a rapid process. You can rapidly mass propagate plants instead of waiting for years to plant millions of the same plantlets. It takes months to generate millions of the same plants, and easy carriage. Like in my room, I can have, if this place is equipped with the material necessary for plant tissue culture, I can have let's say 50,000 plants in this room alone. So, space efficient, just like Henry said, space efficient is time, time saving. And if there is an infection, you can easily detect it. You can easily detect infections. And apart from that, it may also be the solution to food shortage that we have all over the world. Some people don't have enough food. There are many places where food shortage is a lot of problems. So if we can dedicate more time into studying what tissue culture is, and if we can invest in buying buying equipment and materials that may help us carry out effective plant tissue culture, we may be going towards a much more sustainable world for everyone. And then I'm going to give you guys, I think time has gone. I'm going to give you guys the opportunity to have questions. I know some of you will have some questions, important questions that you may want to ask. I'm open to all the questions that you may want to ask. Of course, there are advantages and disadvantages of the process. So I'm open to any questions that you may want to ask. So this is the basic of plant tissue culture. So I'm going to leave the stage to Super Mau and then Mehek for now. Yes.
So does anyone have any question about the topic or something? I think Mao has a doubt, so.
Okay. Okay. Yes.
Yeah, okay. So my question is there like a reservoir or just a collection of plant species in a specific place in which a lot are accumulated so that they can have some sort of inventory?
Absolutely. That's a very beautiful question. Yes. That's a very beautiful. In fact, in the National Center for Genetic Resources and Biotechnology, where I worked, they have what we call seed gene bank. The seed gene bank was designed to keep seeds of any available plants that we have. And they can be kept as long as 100,000 years. And people can come to collect the seeds there. The essence of keeping them there is to make sure that we preserve these species. And sometimes we carry out what we call I've forgotten the terminology. I hope I remember now. Viability test. Brilliant. I remember. Viability test. We want to know if those seeds are very effective. So just like you said, there are people who they take note of the inventories and everything. They say, okay, this seed has been here for 10 years. Are these seeds viable? So they take some part of the seed, they plant them, we propagate them, we calculate their viability rates, the percentage of viability, then we also say, we need to replace them with other ones. Yes, that is very, very, some have it, but we should be looking towards a globalized gene bank, seed gene bank, where we can have, there can be interconnection between nations. That, okay, I know that Mexico has a lot of resources, lot of, plants, animals, natural resources that are not found in many countries. For example, let's say I want to plant nopales, and I want to take that to let's say I want to take that back to England. So, I can ask someone in the seed bank, in the seed gene bank in Mexico, do we have seed of nopales? Does it form seed? Mm-hmm. Does it flowers? Does it make seed? Can the seed be kept? All those questions should be asked. Thank you so much, Maurice. Thank you for that wonderful question. I think we have some questions in chat as well. We have Ivan Aritpe says, are there any method for preparing media at home? Absolutely, yes. We have what we call homemade media. All you need to do, Ivan, I know you are very curious. Ivan is always working on those science stuff in his house. Yes. So Ivan, yes, you can make it. All you've got to do is to buy some of the, for example, you can buy glucose, and you can buy the glucose as the carbon source. You can buy gel. There is a specialized gel. I don't know where they can be gotten here, but you can buy those specialized gel. And also you can buy these minerals and all that. But you need to know, you need to look for manuals online on the percentage, the number of the amount, the mass of the ones, and how to add them together. And also some people do it by trial and error. They do it, they mix this percentage. It's a lot of scientific, you can use the scientific method to do a lot of things there. Yes, it can be done, but it's going to spend you some money, and you have to maybe sacrifice some space in your room, Ivan. Okay. Yes, we have Henry. He said, has this been implemented? You're welcome, Ivan. Has this been implemented somewhere in the world? Yes, Henry, that's a very good question. In fact, it was implemented in Nigeria at some point, but the problem in Nigeria is a very serious problem. There is a problem we call, there's something we call continuity. I'm going to give you an example. The previous Mexican president was Enrique Peña Nieto, if I'm not mistaken. Yes, Enrique Peña Nieto. Imagine Enrique Peña Nieto started the construction of road from Puebla to Veracruz, but he couldn't finish. And his time was over, you know. He said, okay, yeah, to 5 años, se va. And AMLO came. And AMLO says, okay, because this road was not started by me, I'm not going to continue it. That happens a lot in Nigeria. That was just an analogy. I know that that doesn't happen here, but that happens a lot in Nigeria, you know, continuity. That was actually implemented in Nigeria at one time. They invited some specialists from England, from the US, and then, you know, the place is still there, but I don't know how functional it is because, you know, when something is not bringing a lot of money, you know, the idea is not to generate money, is to provide, is to save the nation, is to provide resources for people. And also the monetary aspect is also very important because they need to keep what we call the DNA testing. That is a very, you mentioned an important question, Henry, because there's an important aspect of this, why many people are not into it. For example, you need to have a DNA lab, DNA analysis lab. I'm going to give you an example now. There's something we call somaclonal variation. Somaclonal variation is that changes. It's like mutation. When you culture a plant tissue culture, you know, after some generations, depending on the plant, the genetic constitution changes of that plant. So, you know, you need to consider the stability. For example, for bananas, after 12 generations of planting the same plant that you culture in a specific condition, the genetic constitution changes. So you should have enough equipment to be checking, to be doing DNA analysis, just like we do DNA, excuse me, just like people do DNA analysis to find out someone who is guilty of a crime. It's a lot of work. It's a lot of work. So, and it's a very beautiful process because when you see the result, you have plants of the same size, the same number of leaves, the same height. It's just beautiful. And apart from that, it's a way to, you know, to feed the nation, to provide basic information and resources to the farmers as well, to encourage them. You can give farmers seeds to plant without getting anything from them. That will encourage them to work more, and perhaps they can produce cheaper food for the nation. And also that will help to reduce the concept of food shortage and what have you. So yes, many countries are doing it, but they are not doing it in at the scale that they are supposed to be doing it. I know that in England as well, it's been implemented, but it's not enough has not been done on this to make it very, very, you know, very big. And, you know, that is the idea of talking to you guys. I know that you guys are very curious. Ivan is already thinking on turning his room to a culture room. I know you are very curious. I know that you are going to explore the possibilities out there. So I know that some of you after today may be willing to study something related to this. And let's hope in the future you can take advantage of this brief talk and then to make the world a better place. Thank you so much, Henry. Thank you. Thank you. Great, Ivan. Great. Great. That is where the great ideas are generated, you know, from your thinking, from your, you know, from your curiosity. You never know what you can achieve. You just need to try. Thank you so much, Ivan. Thank you. Yes. I don't know if anyone has a question. Vali Lopez is there. Yes, Jose Emilio Ranque, Badi, Julia Estudillo, Dani Alvarez, Supermao, Ivan, Karen Hernandez, Jocelyn Mesa, Lore Fernandez, Pepe Gonzalez, Henry, Joanna, Shirel, Ike Gutierrez. Yes, Mehek, of course, and Supermao, Mauricio Emilio Monroy Gonzalez.
No, I mean, not from me.
Okay.
If there aren't any questions, I just want to say thank you for your time. It has been an honor learning.
Thank you.
Until next time.
Thank you so much. Thank you. Thank you, everyone.